The future of healthcare, exploring the potential of bioprinting. The concept of bioprinting, a process where living cells and biomaterials are combined to create tissue-like structures, has rapidly evolved from science fiction to a tangible reality in the medical field. As a form of additive manufacturing, bioprinting uses principles similar to 3D printing, but with the crucial difference that the ink consists of living cells and other biological materials. This technology holds the promise of revolutionizing healthcare, with applications ranging from tissue engineering and drug testing to the eventual goal of creating fully functional organs for transplantation. Bioprinting is a multidisciplinary field, combining biology, engineering, and material science. The process involves several steps, including the preparation of bioinks, the design of tissue scaffolds, and the actual printing and maturation of the tissue. The potential implications of bioprinting are profound, offering the possibility of personalized medicine, reduced organ transplant waiting lists, and significant advances in regenerative medicine. This article delves into the current state of bioprinting, its applications, challenges, and the future prospects of this groundbreaking technology. The basics of bioprinting. Bioprinting is fundamentally an extension of 3D printing, but instead of using plastic or metal, bioprinters use bioinks, substances composed of living cells and other biocompatible materials. The process typically involves three main stages. 1. Pre-bioprinting. This stage involves creating a digital blueprint of the tissue or organ to be printed. The blueprint is generated using imaging techniques such as CT scans or MRIs, which provide detailed information about the structure of the tissue. The bioink, composed of cells and a carrier material like a hydrogel, is then prepared for printing. 2. Bioprinting. During this stage, the bioink is deposited layer by layer according to the digital blueprint. The printer builds the structure in a precise manner, replicating the architecture of natural tissue. This process can take several hours or even days, depending on the complexity and size of the tissue being printed. 3. Post-bioprinting. Once the structure is printed, it undergoes a maturation process where the cells proliferate, differentiate, and organize themselves into functional tissue. This stage often involves providing the printed tissue with a controlled environment, including the right temperature, nutrients, and mechanical stimuli, to promote natural growth and development. Applications of bioprinting. The potential applications of bioprinting are vast, with the most immediate impacts expected in the fields of tissue engineering, drug testing, and organ transplantation. 1. Tissue engineering. One of the most promising applications of bioprinting is in the creation of engineered tissues for regenerative medicine. Bioprinted tissues can be used to repair or replace damaged tissues in the body. For instance, researchers have successfully bioprinted skin, bone, and cartilage tissues that can be used for grafting in burn victims or patients with degenerative diseases. In the near future, it is conceivable that bioprinted tissues could be used to treat conditions such as heart disease, spinal cord injuries, and even certain types of cancer. Drug testing and development. Bioprinting also has the potential to revolutionize the pharmaceutical industry by providing a more accurate and ethical alternative to animal testing. Bioprinted tissues can be used to create organ-on-a-chip models that mimic the behavior of human organs. These models allow researchers to test the effects of drugs on human tissues in a controlled environment, leading to more accurate predictions of how drugs will behave in the human body. This could significantly reduce the time and cost of drug development, as well as minimize the risk of adverse effects in clinical trials. 3. Organ transplantation. Perhaps the most ambitious goal of bioprinting is the creation of fully functional, transplantable organs. Currently, there is a significant shortage of donor organs, leading to long waiting lists and high mortality rates among patients in need of transplants. Bioprinting offers the possibility of creating organs on demand, tailored to the specific needs of individual patients. Although we are still several years away from this becoming a reality, significant progress has been made in bioprinting simple organs and organ structures, such as blood vessels, heart valves, and even small-scale liver tissues. Challenges in bioprinting. Despite its enormous potential, bioprinting faces several significant challenges that must be addressed before it can become a routine part of medical practice. 1. Complexity of human tissues. Human tissues and organs are highly complex, with intricate networks of cells, blood vessels, and extracellular matrices. Replicating this complexity in a bioprinted structure is a major challenge. For instance, creating a functional organ requires not only the correct arrangement of different cell types but also the integration of vascular networks to ensure the tissue receives adequate nutrients and oxygen. Current bioprinting techniques are still limited in their ability to create these complex structures, although advances in multimaterial printing and vascularization techniques are showing promise. 2. Cell viability and functionality. Another challenge is ensuring that the cells used in bioprinting remain viable and functional throughout the printing process and beyond. The printing process can be stressful for cells, potentially leading to cell death or loss of functionality. Researchers are working on developing bioinks and printing techniques that minimize stress on the cells and promote cell survival and function. Additionally, ensuring that bioprinted tissues behave like their natural counterparts in the body is a critical hurdle that must be overcome. 3. Standardization and regulation. As with any new medical technology, standardization and regulation are essential to ensure the safety and efficacy of bioprinted products. Currently, there is no standardized protocol for bioprinting, and different labs may use different methods and materials, leading to variability in the quality of the final product. Developing standardized procedures and regulatory frameworks will be crucial to the widespread adoption of bioprinting in clinical settings. 4. Ethical and societal considerations. The advent of bioprinting also raises ethical and societal questions that must be addressed. For instance, the ability to create human tissues and organs in the lab blurs the line between natural and artificial life, raising questions about the definition of life and the ethical implications of creating living tissues outside the body. Additionally, there are concerns about the potential for bioprinted organs to be used for non-medical purposes, such as enhancing human abilities or creating genetically modified organisms. The future of bioprinting. Despite these challenges, the future of bioprinting looks incredibly promising. As technology continues to advance, it is likely that bioprinting will become an integral part of personalized medicine, allowing for the creation of customized tissues and organs tailored to the needs of individual patients. This could lead to a paradigm shift in healthcare, where the focus moves from treating disease to regenerating damaged tissues and organs. One of the most exciting areas of research in bioprinting is the development of bioprinted organs. While we are still several years away from being able to print fully functional organs for transplantation, significant progress is being made in this area. For example, researchers have successfully printed small-scale liver tissues that are capable of performing basic liver functions, and bioprinted heart valves have been implanted in animal models with promising results. In addition to organ transplantation, bioprinting could also play a key role in regenerative medicine. For example, bioprinted tissues could be used to treat injuries or degenerative diseases by replacing damaged tissues with new, healthy tissue. This could lead to significant advances in the treatment of conditions such as spinal cord injuries, heart disease, and arthritis. Another area where bioprinting could have a significant impact is in drug development. By using bioprinted tissues and organ-on-a-chip models, researchers can test the effects of drugs on human tissues in a controlled environment, leading to more accurate predictions of how drugs will behave in the human body. This could significantly reduce the time and cost of drug development, as well as minimize the risk of adverse effects in clinical trials. Conclusion. Bioprinting represents a frontier in medical science, with the potential to transform healthcare by enabling the creation of customized tissues and organs. While there are significant challenges to overcome, including the complexity of human tissues, cell viability, and the need for standardization, the potential benefits of bioprinting are immense. From tissue engineering and drug testing to the eventual goal of organ transplantation, bioprinting has the potential to revolutionize the way we approach medicine and healthcare. As the technology continues to advance, it is likely that bioprinting will become an integral part of personalized medicine, allowing for the creation of customized tissues and organs tailored to the needs of individual patients. This could lead to a paradigm shift in healthcare, where the focus moves from treating disease to regenerating damaged tissues and organs. The future of bioprinting is bright, with the potential to change the way we approach medicine and healthcare. As researchers continue to make strides in this field, it is likely that bioprinting will play an increasingly important role in the treatment of disease, the development of new drugs, and the creation of customized tissues and organs. With continued investment and research, bioprinting has the potential to revolutionize healthcare and improve the quality of life for millions of people around the world.